Good morning, aloha everybody. Thank you for coming to Sunday Assembly. I am your host, Benjamin Yep, and here we love to live better, help often, and wonder more. Spring has arrived, and the world is full of new energy. Before we start, I'd like to point out that this meeting is being recorded in speaker view. That means your video feed won't be recorded and everyone will have muted audio. Feel free to rewatch this meeting on YouTube and Facebook and share it with others who couldn't make it today. Let's all be filled with spring energy and sing along with our SASV band. Stand up if able, clap along, and enjoy the music. Our first song is You've Got a Friend. You may remember singing along to this one last April. It's been a full year since we've been doing these online meetings and singing online. The music team wanted to bring this one back, and Alex is now in it as well. This music video was created by Krista and Elizabeth.
Nothing is going
friend. Ain't it good to know you've got a friend? Ain't it good to know you've got a friend? Oh yeah, yeah, you've got a friend. Thank you, band, for a lovely performance and great video editing. And thank you for everyone for singing at home. Welcome to Sunday Assembly, Silicon Valley. We are here together to celebrate the one life we know we all have. My name is Benjamin, and I'm your host. In 2015, my wife and I moved to San Jose from Los Angeles. We explored many Silicon Valley activities ranging from rock climbing, to late night boba, to improv theater. Eventually, we were attracted to Sunday Assembly's various speakers. Our first event was focused on time travel logistics and if it's scientifically possible to achieve it. We became frequent attendees, joined the SASV community, and eventually joined the band. Sunday Assembly debuted in London 2013. The Silicon Valley chapter is one of a number of chapters around the United States, the UK, and all over the world. Our purpose is to build a community of people that inspire one another to live better, help often, and wonder more. We are a secular community. We don't advocate for any particular religious beliefs, but we do encourage everyone to follow their own inner journey. As the fellow members of the human family, we ask you to join us in adopting the spirit of acceptance and respect. Our local assembly holds this monthly gathering featuring uplifting songs and hopeful, inspiring talks. We also organize small groups, which we call SMOOPs, as a way to build more connections. We aspire to build an open community where we can belong, participate, and make friends. If this is your first time joining Sunday Assembly, welcome! We hope to make you feel home here with us, and air high five! If this is your second time joining, welcome back. We're so happy to see you again. Double air high five. 1, 2. If this is your 10th, 50th time, or even more, it's sure been a wonderful getting to know you over the years, and I'm happy we're spending time together again today. Before we go too far, if you have any questions about how to use Zoom, look to the chat button at the bottom of your screen and someone can help you. Even though you are all muted, we are all still about socializing. During the program, you can cheer by using the sign language gesture jazz hands, and you can use the Zoom reactions for applause as well. Please engage as you are most comfortable. To start, today's creative space will be presented by Krista. April 22nd is Earth Day, and she would like to express her appreciation for our planet.
Thanks, Ben. Hi, everyone. I want to share a music video with you that I put together with the help of Elizabeth, my mom, Ginny, and several musical guests, Sarah Valencia, Kevin Brown, Raul Carrig, Virginia Anton, and singer and songwriter for Sunday Assembly Nashville, Stephen Phelps. Stephen has also graciously done the audio editing for this song, and you will hear what an amazing job he did. The song is For the Beauty of the Earth. It's originally a religious song, which I've written new secular lyrics for in celebration of Earth Month. And it is set to a slideshow of photos that I've taken and others that I've collected. I appreciate that in the Bay, being eco-conscious is pretty common, but there's always more that we can commit to doing. I've started to eat more plants and less animal products in my diet, as they have a huge impact on the environment. And I'm starting to educate Caden on things like greenhouse gases, the rainforest, plastics in the ocean, bees, organic farming, and I love seeing him start to understand and appreciate those issues. If you're looking for ideas on what you can do, you can check out earthday.org/earthdaytips. I hope you can take some time to look for what next steps you can do to further help the Earth. Without further ado, here is For the Beauty of the Earth.
For For the beauty of the earth, for the beauty of the skies, for the love which from above, over and around the skies, over and around the skies, for the earth to you is a joyful song of praise. For the beauty of each hour, of the day and of the night, hill and vale, tree and flower, sun and moon, stars light, sun and moon, stars of light, skies above to you is this a joyful song of praise. For the joy of human love, brother, sister, parent, child, friends nearby, and friends afar, for all gentle thoughts and minds, for all gentle thoughts and minds, for all love to you we raise this a joyful song of praise. For each perfect gift of earth, upon which we do rely, for the air, the land and sea, let us leave a better place, let us leave a better place, for the earth to you raised. This a joyful song of praise. This a joyful song, our song of praise.
Thank you, Krista. That was wonderful. And I feel much beauty in the earth when I look outside. As mentioned, this month's theme is Beyond Darwin. Our speaker today is Megan Hochstresser. Megan has a BA in biology from Brown University and received her PhD from Jennifer Doudna's lab at UC Berkeley in 2016, where she studied mechanisms of CRISPR immunity in bacteria. She joined the Innovative Genomics Institute in September 2016, hoping to bridge the gap between researchers and the public. Megan develops educational resources and programs, spearheading everything from mobile app development to community event planning. She regularly gives talks to students, teachers, scientists, journalists, physicians, and more. As a CRISPR expert and professional science communicator, Megan brings diverse audiences into the conversation about genome editing. Charles Darwin famously presented evolution as a process of natural selection in 1859. In summary, evolution is a directional trait change of a species that normally takes many generations. Soon after, Mendel made breakthroughs to understanding inheritance with his famous pea plants. Then Watson and Crick modeled DNA, a genetic blueprint for humans. Fast forward, the Human Genome Project successfully sequenced the human DNA. Another genetic breakthrough has arrived, CRISPR. What amazing progress science has made in only 150 years. Please welcome Megan Hochstrasser.
All right, thanks so much for that nice introduction, and thanks for having me today. I'm Megan Hochstrasser, and I come from a group called the IGI, or the Innovative Genomics Institute. We're a research partnership between UC Berkeley and UC San Francisco, and we're aimed at using CRISPR and other genetic engineering tools to solve major world problems. So today I'm going to be talking to you about CRISPR, rewriting DNA, and the future of humanity. So everything I'm going to talk to you about today really comes back to one thing, and that's DNA, which I'm showing you here. This is the double helix. This is the blueprint of life that is contained within all of our cells and in every living organism on the planet. DNA is basically an instruction manual that tells cells how to work and how to survive. And DNA controls characteristics about an organism that we call traits. So traits are things like whether or not you have dimples, the color of your eyes, things like that, that I've stolen here from 23andMe. So if you do 23andMe, you can test to kind of understand the genetic basis of some of these traits that you presumably already know that you have. And that can be kind of fun, but there are also sort of less frivolous traits that you can have, like genetic diseases. So there are a variety of different genetic diseases. There's actually thousands of them. I've taken, again, just a sampling here from what 23andMe tests for. And you can tell that I'm sure most people have never heard of a lot of these. I've never heard of most of them because there can be just a handful of people around the world that have a particular condition. Or they can be more common, like a lot of people have heard of cystic fibrosis. That's technically a rare disease, but it's a little bit more common than some of these other really, really rare conditions. And so these are the kinds of conditions that we're learning more and more about, and there's more and more resources for being able to test for them. But until pretty recently, you could read these sequences, but not rewrite them. So you would have no way of actually changing any of these sequences or correcting mutations. And that's why people are so excited about CRISPR, among other reasons, because CRISPR is a tool that lets us change DNA. And it makes it a lot cheaper and easier than prior tools, which I don't have time to get into, but can answer questions about later if you like. So CRISPR is usually characterized by this protein here shown in green called Cas9. So that's CRISPR-associated protein 9. And that's the main protein or enzyme that we use to do all of the genetic engineering that we do. So what I'm showing you here is an artist's depiction of what this might look like at the microscopic level. And I think it can be kind of confusing to think like, how can we as non-microscopic human beings control what something microscopic is doing? And it all comes down to this little piece of RNA. So the importance of RNA is more recently understood because of the RNA vaccines that are out there. But in this case, we can program this protein to cut and change wherever we want in DNA by changing this little piece of RNA that it carries. So how exactly does that work? If we want to program Cas9 to go edit at a new place in the genome, first you have to pick your target. So you need to know, I'm showing a target in blue, for example, what sequence you want to change. So that might have a mutation in it that's causing a disease. There might be other reasons. So we need this sequence here. And then the next step is to give Cas9 that same sequence. So we basically give it a location code or a zip code encoded in this thing called the guide RNA. So there's part of it that is all twisted up and forms these things called stem loops. That's like the handle that this protein hangs onto. And then this part right here, we can change this. So as scientists, we can order this piece of RNA and change just this part. And that's what tells the protein where to go. So when Cas9 finds its target, it does this by unwinding the two strands of DNA and coiling the guide RNA into the DNA. So it basically makes its own little double helix by pushing out one strand of the DNA. So this is the nitty-gritty molecular detail of how things work. Once it is able to find its target, it makes a cut. So why would you want to make a cut in DNA? That sounds a little bit counterproductive, maybe, but it's actually the first step in editing the genome. So CRISPR, you can just simplify it down to being a pair of scissors that lets you cut at a place in DNA. So once you do that, which is called a double strand break, this sends the cell into freak out mode. So the cell needs to repair this gap or it'll die. And luckily people, humans and plants and everyone have all these kinds of repair systems built into our cells because we're always getting damage to our DNA from the sun and from errors, all kinds of things. So these repair systems go to work at this break and they can fix it in one of two main ways. One of them involves kind of frantically slapping the two ends together, throwing in a few nucleotides, taking out a few. And this sounds kind of bad, but it can actually be useful because imagine you're ripping out pages from the middle of an IKEA instruction manual. Imagine the furniture you'd make at the end of that. It would be probably kind of messed up, right? So that's actually really useful if we're doing scientific research, because if we want to understand what a gene does, breaking it is a really good way of getting clues. The other way that we can resolve this cut is by adding in a piece of donor or template DNA. So if we, as the scientists, along with the CRISPR tools, throw in a piece of DNA, we can sometimes get the cell to paste that in over this gap and insert a new piece of DNA. So this is what we want to do most of the time if we're trying to fix a mutation. So just to kind of summarize all of that in video form, since I know molecular details can be confusing, I'm just going to play this briefly. So we're going to go inside of a cell here, and we're specifically going into the nucleus where all the DNA is stored. So there's going to be multiple Cas9 CRISPR molecules searching around this DNA, looking for their one target that we've programmed. And this next one's going to actually find its target. So it's going to grab onto the DNA and slowly start unwinding the two strands. And then the guide RNA that matches that DNA is going to base pair and wrap up. So once it finds the target, it's going to make a cut, like you just saw. And then other proteins, like the repair proteins, are going to bump it out of the way and fuse those two ends together again. And you can see in this example, we added these two little magenta pieces of DNA or genetic letters. So in that way, we made a change at that site. So that's how gene editing works. I like to kind of summarize it at this point, since this is not super intuitive for everybody. I think a good way of thinking about it is imagining if you're editing a sentence. So if you had a typo in a sentence like this, we could use something like CRISPR to fix it and restore it to the song lyric that we want. So text editors are a good analogy for CRISPR. Sometimes people also describe it as genetic surgery, if you think of it as cutting out mutations and replacing them with healthy copies. So just now, CRISPR lets us change DNA. Why is this powerful? Well, if something has DNA, that means we can edit it now. And what I'm showing you on the screen here are so-called model organisms that have been the main workhorses of scientific research for decades and decades. So it's really common to study things like fruit flies, yeast, mice. And in large part, that's because we have been able to change their DNA with these really specific methods developed just for them. And that's great. We've learned a ton, like all of modern genetics is based on studying these guys. But there's so much diversity of life on Earth. So with CRISPR, we've now been able to edit all of the organisms that I'm showing you here and many, many more. So that means we can now do things like study how butterflies pattern their wings and how that's controlled by their DNA, or how why mushrooms turn brown after you've cut into them after a while. And we can potentially change that so that they don't turn brown as quickly. So all kinds of things are kind of now on the table to be studied using CRISPR, which is very exciting. There's a lot of other reasons to be excited. So beyond basic research and medicine that I mentioned, we can also do a lot in agriculture. So our institute, we were just having this workshop last week about how we can potentially impact climate change. So we're doing a ton of planning to see, can we mitigate the effects of climate change? Can we give farmers crops that will be drought resistant or can otherwise withstand the effects? Can we engineer microbes to capture and sequester greenhouse gases and kind of take carbon out of the atmosphere? So there's so much stuff we can do. We're no longer as bound by our tools. It's more, what is the limit of your imagination? And so I just want to give one example of an exciting medical application. So sickle cell disease is one of the first conditions that's being treated using CRISPR. And if you're not aware of sickle cell disease, it's a genetic disorder caused by a single typo in DNA, just one letter out of the millions causes this awful condition. And it causes red blood cells that usually would look like this one, this nice kind of round one. It distorts them into this sickle or half moon shape. And so when your cells are like that, they can't really get through blood vessels, they get clogged up. And this is a really, really painful disease that shortens the lifespan and causes all kinds of damage. It's really awful. And so this makes it, because it's just this one typo, it's actually kind of good for CRISPR. It's a good target for CRISPR because we can take this single nucleotide or this single letter that's wrong, potentially use CRISPR and correct it to the right one and potentially restore cell function. So this is not just science fiction, it's actually been done. So there's this really interesting series on NPR that I would encourage you to look up if you're curious about this, following this first patient in the US who's gotten this sickle cell treatment. Her name is Victoria Gray, that's who's pictured here. And she's been very open about the experience. And this was done, I think, a year or two ago now, and she has not had any pain crises. She's not required blood transfusions. It seems like she's essentially, for the time being, at least cured of the disease, which is really major. And again, I'm very hesitant to say cured because this is something where we need to monitor her long-term, make sure there aren't side effects, make sure things don't, go downhill for some unexpected reason. But it's incredibly promising so far. And if you're interested in seeing kind of what's going on in general in clinical trials land, we did a really nice update recently on our site where we summarize all of the different trials that are happening now. There's maybe like 5 categories of trials. And this includes treating a particular form of genetic blindness, this amyloidosis condition that you can treat by editing the liver, sickle cell, beta thalassemia, things like that. So if you want to learn more, I would check that out. So now that we have so much power, we of course also have a lot of responsibility. So now that we can do so many things, it comes down to deciding which of the things we should be doing to really maximize public benefit and ensure that these benefits are distributed equitably. So one of the issues that people talk about sometimes is GMOs. So GMOs, are genetically modified organisms, are the product of a previous form of genetic engineering technology, not CRISPR. And there's all kinds of backlash and social argumentation and everything about GMOs that CRISPR is kind of now walking into. And so there's a lot of discussion about how this technology is different, how we might be able to benefit people using it and not get pulled into these same debates that GMOs have gotten pulled into. And that's such a huge topic. I could give an hour-long talk about it. I'm happy to talk about that later with anybody. But I would say that scientifically speaking, there's nothing wrong with GMOs. There's only potentially kind of social problems with them. So we'll see where CRISPR goes, but there's a lot that can be done agriculturally. There's another use of CRISPR called gene drives that I can't even begin to explain. It's really complicated, but one of the ways that we could use gene drives would be to wipe out an entire population of mosquitoes, like a whole species. It's a kind of crazy technology that sounds, potentially ecologically terrible to do something like that, but then you also have to think these mosquitoes might carry malaria, and that has such a human toll of suffering and death that comes with it. How do you decide whether, inaction means letting people die of malaria versus taking an ecological risk? There's a lot of kind of interesting stuff to think about there. And then last, probably the biggest ethical issue that people talk about with CRISPR is the potential to genetically engineer future generations. So you could engineer sperm, eggs, or most likely an embryo during the process of IVF or in vitro fertilization. Imagine not just adding sperm into the egg, but adding CRISPR. You could potentially change someone before they're born. And this introduces so many ethical questions that I can't get into. I can talk about that in the breakout rooms if you like. I'll just mention that this did happen. So in China, a couple of babies were born that were CRISPR engineered, and it kind of sent up red flags across the world. So there's now a lot of efforts by people, by oversight bodies, to make sure that this doesn't happen again in the way that it happened there, and that we discuss ways of potentially governing and regulating the technology worldwide so that people only do what matches societal desires. Okay, so if you want to learn more, there's now even more books about this. This new one from Walter Isaacson just came out. There's a really good documentary that's now on Netflix called Human Nature. I really recommend that. And if you want to kind of keep following the technology, we post about it all the time on all different kinds of social media. So feel free to follow IGI. And if you want to keep up with me, I'm on Twitter at the Chrispress. All right, thank you.
Thank you, Megan. That was fascinating, and I've learned so much. To show our appreciation, Sunday Assembly will be donating $50 to end the backlog. For everyone else, please hold any questions you'd like to ask Megan for the speakers table at the end of Assembly. I know myself have a lot of questions because she lit up a whole bunch of light bulbs in my mind. Now, what's better than one song? Two songs. Even though superpowers aren't here today, we can still sing about them. Please enjoy Something Like This, presented by our band. This music video was edited by Elizabeth.
I've been reading books of old, the legends and the myths, Achilles and his gold, Hercules and his gifts, Spider-Man's control, and Batman with his fists. And clearly I don't see myself up on that list. She said, where'd you wanna go? How much you wanna risk? I'm not looking for somebody with some superhuman gifts, some superhero, some fairy tale bliss. Just something I can turn to, somebody I can kiss. I want something just like this. Do-do-do-do, do-do-do-do, do-do-do-do. Oh, I want something just like this. Do-do-do-do, do-do-do-do, do-do-do-do. Oh, I want something just like this. I want something just like this. I've been reading books of old, the legends and the myths, the testaments they told, the moon and its eclipse, and Superman unrolls a suit before he lifts. But I'm not the kind of person that it fits. She said, where'd you wanna go? How much you wanna risk? I'm not looking for somebody with some superhuman gifts, some superhero, some fairy tale bliss. Just something I can turn to, somebody I can miss. I want something just like this. Oh, I want something just like this. Oh, I want something just like this. Do-do-do-do, do-do-do-do, do-do-do-do. I want something just like this. Do-do-do-do, do-do-do-do, do-do-do-do. Where'd you wanna go? How much you wanna risk? I'm not looking for somebody with some superhuman gifts, some superhero, some fairy tale bliss. Just something I can turn to, somebody I can kiss. I want something just like this. Oh, I want something just like this.
Thank you, music team. That was super. Next in our segment called Doing Your Best. And a community member shares a personal story of how they are doing their best. Today we have Marshall.
Hi. Aphasia is frustrating. I got aphasia when I had my stroke 11 years ago. Every year, more than 250,000 people in the United States are diagnosed with aphasia. Aphasia is more prevalent in Parkinson's disease, muscular dystrophy, cerebral palsy. The National Aphasia Association found that 84% of people have never even heard of aphasia. Between 25 and 40% of stroke survivors have some form of aphasia. Aphasia is a neurological disorder that results from damage to the language center of the brain. People with that aphasia may have difficulty with speaking, understanding, writing, reading, and math. I am reading this because otherwise the talking will suffer and be unintelligible or halting. Other people with aphasia are mistaken for being well out intellect, but sometimes others may not understand their intellect is still intact since they had difficulty with language. I was on safari in Tanzania when my cough got so bad that I couldn't even go on the Jeep. Fortunately, I got some better and was able to join the group. I returned home and played golf in size 78. The following day, I was told I drove myself to the emergency room and was admitted. I had suffered a tear in a carotid artery. I spent 6 weeks in the hospital, but I don't remember much about my time there. My brother flew in from Denver and was unaware of the serious nature I had. About a 50-50 chance of my making it. Since then, I gotten divorced, live by myself in a condo. I can drive, fortunately, and have a scooter which takes me around shopping. I miss my piano tuning very much. 4,000 clients. I have tuned for Liberace and the Pope. I am very fortunate to have a network of friends. I spend most of my time in Zooming classes without regard to location. New York City, Canada, Utah, and Oregon, just to name a few. Will I get better? Who knows? But I am trying.
Thank you, Marshall, for sharing your story. If you want to be part of our celebration of Sunday Assembly and do a segment like reading or doing your best, just email at the address in the chat. We will find a role for you in the program that is just right for you. We'd like to hear from you all, especially about what inspires you to live better, help often, and wonder more. Next, Bill has a message for us from the Sunday Assembly Silicon Valley Board of Directors.
Hi, folks. My name is Bill Hathaway. I've been attending Sunday Assembly since somewhere in 2014, and I've been a member of the Board of Directors since this past July. And while we really appreciate everyone's attendance and enthusiasm, and of course donations, the main resource that keeps Sunday Assembly running is volunteers. And we're looking for 3 types of volunteers. So if any of these areas sound interesting to you, please let us know. The first area is people who are willing to volunteer for a segment of an assembly. And these can include doing a reading or the creative video that Krista produced for us today, or doing your best segment, such as we just heard from Marshall. The time involvement for doing something like this is often about an hour or so. The second area that we're looking for people to help out on is the action team. This is a group that meets every month to plan our upcoming assemblies and helps to run the assemblies, whether it be tech, it could be helping us find speakers, and other like maybe new SMOOPs that we're going to do. The time commitment for the action team is anywhere between like 2 to 5 hours a month, depending on like what responsibilities you would like to take on. And then there's one more area which you've been hearing about a little bit recently, but is often behind the scenes, which is the board of directors. The board of directors has a monthly meeting, and then some of the current board of directors have been on it for over 2 years, and we really like to be able to give them a break. So we'd love to talk to anyone who is interested in being on the board, and especially if there's anyone that has nonprofit board experience, that would be wonderful. The current board consists of myself, Krista, Fausto, and Richard. We'd like to thank Raji and Jean, who are stepping down or in the process of stepping down. And I'd also like to give a big heartfelt shout out to Jean for her work as treasurer of Sunday Assembly. And that is a role that I am now taking over. And then Richard is also assisting me with that. So if you're interested in volunteering for any of these things, we would love to talk to you about it and see if there is something that's a fit for you. So please feel free to reach out to me or anyone else on the board. Thank you.
Here at Sunday Assembly, we promote mindfulness, a moment to meditate and self-reflect on your current state of being. What are your emotions? What are you physically sensing? Clear the mind and focus on the now. Steve has prepared an audio focus with the sounds of nature from our local Henry W. Coe States Park near Gilroy. This recording was made in the morning after a rainstorm in a meadow overlooking a wild section of the Upper Coyote Creek at 7:16 a.m. on March 20th, 2021. The wildlife we hear is distant and sparse, and the site is not far from the city of Gilroy and Highway 101 to completely evade the background roar of civilization. Please join us for 2 minutes of meditation. Welcome back, and I feel refreshed. How about you? We're about to do the Life Happens segment where we share updates about our lives. They're whatever you want to share triumphs, difficulties, struggles, joys, whatever. If you haven't shared your life update yet, please feel free to do it now. There's still time to put it into the chat right now. Our lives are full of events, both pleasant and unpleasant. Sharing heightens our joys and makes our pain easier to bear. At times we face challenges that overwhelm us. A real community understands that we aren't perfect, and that our lives aren't either. That's why we strive to make this community a place where we can share difficulties as well as joys. A place of compassion, where no matter what you're going through, you are welcomed, accepted, and even loved. So now here's Richard to bring us all of our news.
But first I have to unmute. Okay, we look as if we have a pretty full docket today. This started with an item from Gordon. Our son and his partner got vaccinated. And then from Lisa and Gordon together, our son Aaron and his wife Savannah are coming home and staying with us for a month. They're in grad school in Connecticut, and we haven't seen them since Thanksgiving 2019. They arrived last night, and we are over the moon excited. So anybody who wants to give a clap for that piece of good news. Mark Robichek, whom you'll be seeing soon, celebrated his birthday on March 25th, and the medical community gave him a gift, a COVID shot. Ben, Iko, and Cedar have an important piece of good news. Baby Cedar met another big milestone and started walking last week. He's now toddler Cedar. Yeah. Raji has good news too. I got the jab this week. Johnson & Johnson vaccine. One and done. Woohoo! Matt Lockwood, over from Sunday Assembly London, says, I'm not sure that it's really news, but it's the 8th anniversary of my first Sunday Assembly. A fun, exciting journey of tears, cheers, and several beers. Well, they do their assemblies differently over there. From Lindsay, I took advantage of education being eligible for the vaccine and got my first shot of Pfizer a week ago. Yay! Lindsay also turned 39, and Lindsay says, I'm teaching my distributed systems course publicly on Twitch this spring. If you want to learn about distributed systems, tune in at twitch.tv/Lindsay Cooper. You know, that takes me back about 40 years. I'll have to think about it. Raji also has a piece of sad news to report. A dear friend who was an amazing woman and role model died fairly suddenly a week ago after a severe stroke. I'm sorry to hear that. I know it's always terrible to lose a friend. David reports, I'll be turning 38 this month. Krista reports, Caden's crib has finally been converted to a bed after he started climbing out. He's doing a great job staying in bed until his alarm says it's time to get up, thank goodness. Whoa, but can he read the clock face yet? Because my kids couldn't. Andre, an old guy coming back after a while, has bought a keyboard, and if my spreadsheet will show me this, is starting learning how to play piano using Simply Piano. Well, keep it up, Andre. I have heard that is difficult. Bill and Cleo report that they both got vaccinated. Lincoln got his first COVID shot. A few more weeks and he can emerge from hibernation. And Elizabeth, this is not the Elizabeth who sings in the band, but a new Elizabeth, reports, we are moving from Louisiana to Palo Alto in May. So welcome to the Bay Area, Elizabeth. And to wind up, just 3 days ago on Thursday, I got my first shot of Pfizer, and I only had to drive an hour to get there. That's all our lives. Back over to you, Ben.
Thank you, Richard. It sounds like the vaccine is the most popular announcement that everyone is happy to have. Time for more announcements. We don't only assemble once a month, we get together in a wide variety of small groups, or SMOOPs, throughout the month. Here's Mark to tell us what's happening. Mark is muted, so give him a moment to unmute.
Oh, sorry about that. I'm a Zoom pro, I should know better. Anyway, good morning, everyone. First, I want to thank Greg Coladonado. We have been way behind on getting our assemblies posted on YouTube, and Greg has caught us up. So thank you very much, Greg. Now, our next SMOOP is going to be on Monday. That's tomorrow at 7 p.m. Compassionate Listing, hosted by Raji. And next comes Happy Hour. This is hosted by Gene and Steve, and it'll be this coming Friday at 5:30 p.m. Next comes Keep Coyote Creek Beautiful. This is a creek cleanup event organized by Keep Coyote Creek Beautiful. This is an Earth Day creek cleanup titled Can It for a Greener Planet. The event happens from 9 to noon Saturday morning, April 17th, but most Sunday assemblers will be there at 10 a.m. Then the action team that Bill mentioned, the action team meets on Sunday next week at 9 a.m. This is a group of volunteers who help by planning upcoming assemblies and occasionally other events. Everyone is welcome. Then on Wednesday, April 21st, hosted by yours truly, Scattergories, a way to have some fun and keep your brain oiled, well oiled. And we have the SASB board meeting. This happens on Thursday, April 22nd at 7 p.m. This is a business meeting, not a social hour. And then a fun one. It's a sing-along SMOOP. This is anyone is welcome. This is from 3 to 5 p.m. on Sunday, April 25th. And next, we want to introduce a brand new SMOOP. This one's hosted by your host for today, Ben. It's a new Pictionary game night. This is going to happen 7 p.m. Wednesday, April 28th. And then we have a community building brunch. Once again, anybody welcome. This happens Sunday, May 2nd at 11 a.m. Now this one, I'm going to introduce Fausto. He's going to talk about this particular Help Often SMOOP. So Fausto, you take it.
Yes, so this is also fairly new. We started this in February, and the purpose is to find ways to help each other within Sunday Assembly, but also do good in the neighborhoods we live in. So for example, the creek cleanup on April 17th came out of this, the last month conversation. So if you want to contribute to new ideas on how to help often, come join us on May 3rd. Back to you, Mark.
Now, our next assembly happens on Sunday, May 9th. You may notice that happens to be Mother's Day, but it's not just Silicon Valley that has a Sunday assembly. Let's visit some other Sunday assemblies. So here's a list of all the times and dates that they happen, and these times are all in Pacific time. Now, before I turn the floor back to Ben, how about a joke? A priest, a minister, and a rabbit walk into the Red Cross to donate blood. The nurse asks the rabbit, "What's your blood type?" The rabbit answers, "I'm probably a type O." And with that, back to you, Ben.
Thank you. That was very fun to hear. And I hope to see other people at the Smoops. Today, so many people helped us to make our meeting happen. We have our thanks to our presenter, Megan Hochstresser, created space by Krista and Elizabeth and Ginny, and all of the musical guests. We have Doing Your Best by Marshall, all of our musicians and singers, and of course, the organizer to make everything behind the scenes happen, which is going to be Steve Sargent and Bill Hathaway today. And then our writers of our newsletter and media, and those who contribute to Life Happen. And most of all, and very much importantly, thank you to all of you who are in attendance. Your presence is what makes our Sunday Assembly all the warmer. I reflect on the amazing science that has come in just the last 150 years. There's the telegraph to smartphones, horseback to Mars space travel, dark, dark nights to entire cities powered by electricity. And as we discussed today, we have accepting evolution to advancing gene manipulation and possible medical breakthroughs. Wow. The mantra of a famous superhero is, with great power comes great responsibility. Knowledge is power, and we must all be responsible for how we affect the future. April 22nd is Earth Day, a day to remind us that our planet needs protection. CO2 emissions, pollution, needless waste are byproducts of technology advancements. Please do what you can to reduce your carbon footprint. If you want to stay in touch with us between meetings, we have a chat email group. You'll find the address to subscribe in the Zoom chat stream. Now we are on to our last song for today, which is What a Wonderful World, presented by our band. My wife and I, Aiko, edited the video.
I see trees of green, red roses too. I see the moon for me and you, and I think to myself, what a wonderful world. I see skies of blue and clouds of white, the bright blessed day, the dark sacred night, and I think to myself, what a wonderful world. The colors of the rainbow, so pretty in the sky, are also on the faces of people going by. I see friends shaking hands, saying, how do you do? They're really saying, I love you. I hear babies cry, I watch them grow. They learn much more than I'll ever know, and I think to myself, what a wonderful world. Yes, I think to myself, what a wonderful world. Oh, yeah.
What a wonderful band for making that music video. And thank you everyone home for singing along. As you enjoy the rest of your Sunday, please join us and shout out the Sunday Assembly motto: live better, help often, and wonder more. This is now the end of our main program. So long and thanks for all the fish. You're all welcome to stick around and join us at our conversation tables. Don't forget that Megan will be available for questions at our speakers table breakout. Zoom doesn't let us mingle quite like the way we would in person, but we're eager to help you out. If Zoom doesn't let you self-navigate, just leave the breakout room you're in and ask to be moved to another table. If you're looking for an icebreaker in your new breakout group, discuss what superpower you would want to have and describe a situation it would be applied.